Hello, everyone. I'm fairly certain that you're aware that mechanical thrombectomy completely transformed the way that we treat stroke patients today. The ability to remove the clot mechanically instead of trying to dissolve it chemically doubled the number of patients that will survive their stroke with no or very limited disability and reduced the number of patients with abilities for life requiring assistance by 30%. That means that today, from a stroke, 5 out of 10 patients will come out with a good outcome, and 4 out of 10 patients will require assistance for life. And it's right here where we can do better in order to reduce the burden of stroke. Now, how can we do better? The best way to do it is by increasing what is called the first pass effect, which is the capacity to remove the clot in a single mechanical thrombectomy pass. Unfortunately, today, only in about 40% of cases are we succeeding in achieving a first pass, while in the other 60%, basically each consecutive pass will reduce the patient's chances of having a good outcome by an average 20%, increase the hospital costs by up to 30%, and the annual care costs for those patients by up to 27%. Now, in order to achieve a first pass, the physician needs real-time information, and the only way to get that today is through the angiogram, this grayscale imaging that is perioperative. But that leaves many questions open. What is the length of the clot? What's its heterogeneity? What's its composition? Is it actually an embolus, or is there an underlying disease that is the origin of the stroke? And that, despite the fact that there's overwhelming evidence in the literature showing that all of these different factors are actually impacting the mechanical thrombectomy procedure, which means that missing information can be the difference in between a patient that walks out of the hospital after 72 hours and a patient who stays in hospital for 6 days or more and will require assistance for life. So if we want to do better, we just need more than the angiogram. And this is exactly what Sensome is proposing. We want to bring color into the grayscale angiogram, providing the physician with information about the clot characteristics, its length, its heterogeneity, its overall composition, allowing the physician to basically make an informed decision whether he just uses aspiration to get that clot on the first pass, or if it makes sense to add a stent retriever in the procedure in order to achieve the first pass, and with that maximize the chances of the patients to have a good outcome. To do this, we've built a unique technology based on electrical impedance spectroscopy. So we've basically taken this shoebox-sized box, an impedance meter, and shrunk it down to a degree that you can actually integrate it into an evasive medical device, creating the smallest impedance-based tissue sensor in the world. This sensor, connected with our predictive models, allows us to identify the tissues that are in contact with the sensor, and that is information that we can then provide to the physician. The first device to integrate this technology is called the Clot Smart Guide Wire, an 014 wire that allows you, like any other guide wire, to access the clot. And on top of just accessing the clot, it allows you, using electrical impedance spectroscopy, to scan that clot. This information is then sent via a transmitter to a tablet, and it's on this tablet that the physician then has the information about the clot characteristics. This integrates seamlessly into the current workflow. There's no additional step. The physician just uses, instead of his regular guide wire, the smart guide wire Clotilde. Now, Clotilde really is only the very first device that we are building, as we've built the world's first smart platform for minimally invasive medical devices, allowing to equip them with our sensor, comprised of 3 parts: the world's smallest impedance sensor, our proprietary sensor technology integration processes that allow us to put the sensor into the device, and our proprietary predictive models that allow us to identify the tissues in contact with the sensor. These go beyond just characterizing clots. We've also been able to demonstrate that we can identify cancer cells and such tumors, which opens the opportunity to pursue indications that are beyond ischemic stroke alone. For example, lung cancer biopsies, when we want to diagnose lung cancers, and beyond that, peripheral vascular procedures where we want to detect fresh clots, and many others down the road. Now, in all of these 3 indications, we're actually already at the clinical stage. In our very first in-human trial for stroke, we've been able to demonstrate that our device is safe in 41 patients and have been able to show that our device performs in situ. Beyond this, we are currently active and about to be finalizing a trial in lung cancer biopsy, and just last year finished a trial in peripheral vascular intervention with very positive results. Now let me present you the team that is behind these developments. We are an international team of about 30 talents that are working hand in hand together from different backgrounds such as microelectronics, biology, and data science, and they work together with the leading physicians of their respective fields to develop devices that actually matter for their patients. With all of these pieces in place, we are tackling some very attractive markets. The first one is that of neurovascular, where basically we have a lot of sprawling M&A activity as early as the R&D phase, because we're looking at a market that is chronically underpenetrated, with only about 12.5% of patients that actually could be treated, meaning that we have up to 2 million eligible procedures per year, representing for Sensome a business opportunity of up to $3 billion. The lung cancer market is a market that is currently being disrupted with the arrival of minimally invasive treatment paradigms as well as robotics. All over the world, screening programs are being put in place, which means that the same number of physicians are facing a steadily increasing number of patients that need to be diagnosed, meaning that the biopsy procedure needs to become more and more effective. Estimates show that we are looking at least 1 million eligible procedures per year, meaning for Sensome technology, about $1 million market. And finally, the peripheral vascular market, which has seen a lot of activity over the last 18 months, because we're looking at a market that is slowly starting to consolidate. Again, the reason is we are only treating about 15% of all the patients that could be treated, meaning that we are looking at a potential of 2.5 million eligible procedures, and for Sensome technology, a $3 billion market. Now, if all of this has sparked your interest, then please join us in this revolution, as these institutional investors have already done, as well as industrial partners such as Asahi Intech, who are building and manufacturing our smart guide wire, or Cosmotech, an M3 company that will be distributing our device in Japan. Please join us in our mission to bring connectivity and real-time information to minimally invasive medical devices, enabling a more personalized care approach that will benefit patients and the healthcare system. Thank you.